life actually exists, but life started about a couple billion years ago, and one fundamental question of biology really is how the heck did it all get started? So that's what I'm going to talk about with you today, and hopefully you guys had a chance to talk about these two questions here. There really are two ways of approaching this question, and one of them is take a modern cell and say, what parts can we get rid of and still have something that's alive? And the other way to look at it
Yes! I love it.
Start from the bottom up and say, what do you need to put together in order to have something alive? And so, if you look at a really complex modern cell like a plant cell, what parts can we get rid of?
None.
Go. What can we get rid of?
So smooth ER.
Okay. Maybe you don't need smooth ER?
I don't think so.
Are there organisms that don't have smooth ER?
Are you asking me?
Yeah, absolutely.
I'm pretty sure there are.
Yeah, go.
Cell wall?
Yeah, you can get rid of the cell wall. Animal cells don't have cell wall. What else? Yeah, animal cells don't have cell walls. You can
I
get
feel
rid of
like you
that.
can't though.
You don't need that.
He's not saying if you took it away from that cell. He's saying of all cells, which are the only necessary parts.
Oh, okay.
Exactly.
Okay.
Which is the things we can get rid of. You don't need a chloroplast. We don't have chloroplasts. Go.
Nuclear membrane?
Don't need the nuclear membrane. Bacteria don't have a nuclear membrane. So you see, we can really delete a lot of these things until all we really need is a barrier, some kind of membrane, and then some kind of heritable material. It's really at the barest minimum what you need to have to be alive. So you got to be able to separate inside from outside. You got to have some information molecule. And then if you build from the ground up, what does it need to be able to do? Well, it has to be able to get bigger in size. It has to be able to make more of itself. It has to be able to do some energy processes, which is what metabolism is. And it has to be able to change heritably over time. It has to be able to compete with other things. So I want to focus on the membrane today. And you all know that modern membranes are phospholipid bilayers with a bunch of proteins stuck in it, right? And that's a really complex structure if you think about it. But let's just take a look at the phospholipid. What do you know about phospholipids and, say, transport of water across membranes? How does water get across modern membranes?
It just can.
Does it just kind of squeeze its way through?
You need like
Yeah, what kind of protein?
Aquaporins.
You need aquaporins, okay? So, you could maybe do better if you could get rid of aquaporins and still have some kind of transport. So let's look at this and say, is there any way that we can simplify this molecule and still have something that'll form a bilayer?
Are you asking, like, could we cut off the bottom?
Is there any part of this that we could get rid of, simplify it?
Totally.
Yeah. Catherine knows.
Get rid of one fatty acid tail.
Okay, we'll delete off the fatty acid tail.
That's a good idea.
And maybe even get rid of this.
Yeah.
Right, so we got this, okay? So this thing right here is just a fatty acid, and it has a hydrophilic part and a hydrophobic part. So if you stick this in water, it will spontaneously form bilayers and globules of fat.
Mr. Maxwell, how long did it take you to cut that out of the image with just your left hand?
Well, I did this beforehand.
Yeah, because we had this
Beforehand?
I did this beforehand. Yeah.
Unfortunate pun.
These guys will spontaneously form, and even cooler, fatty acids will form spontaneously without any biological thing around hydrothermal vents. So simple fatty acids will form out of those conditions. So it's plausible that you could get something from non-living to a lipid layer. So they'll spontaneously form micelles, which are these guys here. Micelles are the ones without a cavity in the center. And they form vesicles, and they're just basically bilayers with a hole in the center. Okay? And so we'll call these, I'll probably switch back and forth between calling these vesicles and calling them protocells, but just know that I mean the same thing. Okay? Okay, so the paper that that this is all coming from is one that was written by Adamala in the Shostak lab. And the real question they're asking is, if you stick a simple catalyst in a vesicle, can you get that vesicle to have competitive advantage? And this was published in Nature Chemistry back in May. So what's a catalyst? What does a catalyst do? What's a catalyst?
Starts reacting.
Speeds up.
Speeds up reactions, okay? Biological catalysts. What are biological catalysts?
Enzymes.
Enzymes, okay? And RNA can actually serve as an enzyme. They call them ribozymes. But so a catalyst is just typically a protein, and the simplest form of a protein you could possibly get, you get these big, complex, big proteins, break it down to two peptides, a dipeptide, that's the simplest you can get. And this is a dipeptide that serves as a catalyst, okay? And basically it takes that thing there, attaches it to that thing, and you get this thing. And then this thing is actually hydrophobic, and it'll localize to the membrane. So because this localizes the membrane, that pulls the reaction in this direction. Does that make sense? Okay. Okay. So, back to our conditions for life. We need a barrier, we need to grow, we need to be able to multiply, divide. We need metabolism, and we need to be able to compete. So that's our big picture. Okay, now I'm going to show you a couple pictures here, and I want to orient you to what they're actually doing in the experiments. Basically, you take two tubes full of liquids, one of which has a vesicle that you label with a dye, and then the other one doesn't have any label. You put them together, and then you see which one, you see the average diameter of vesicles with dye, okay? And so you do this experiment, and then you do the reciprocal, which you dye the other thing. And then you see, because you want to make sure that the dye doesn't actually have an effect. We good with that? Okay. So this, wrong way. So what they did here is, here's vesicle A, and this is what they put in vesicle A, and then vesicle B, what they put in vesicle B, and then they look at average diameter and of the things, and then the number they use is the final over the initial. Now, if there's no growth, that number should be what? 1. Okay, so you can see there's no growth here. If you put a vesicle with catalyst and a vesicle without catalyst vesicle with catalyst against itself, there's no net growth. If you put a vesicle with catalyst and a vesicle without catalyst together, you get growth. Okay, and if you dye the vesicle without catalyst, you get shrinkage. Right? So they do the reciprocal just to make sure the dye doesn't matter. But you can see that with catalyst, you get growth, and this is a second catalyst that I didn't show you, but another catalyst, you get growth. Okay. Then they looked at a similar type of thing, only they fed they have a competition here, and they feed them micelles. Remember, micelles are just globules of fatty acids without a center. And then they reacted. So here we fed them micelles, and you can see against itself, this is your control. And so SH against something, so catalyst against the vesicle without catalyst, you get more growth. So catalyst against nothing, you get growth. Okay? So they're competing. Go.
Just out of curiosity, when you take the micelle and you combine it with the vesicle, so the vesicle is the bilayer, the things go towards in the direction of bilayer as far as stability?
The vesicles, yeah. The vesicles will fuse with the bilayer and become part of the bilayer.
I guess it would seem to me like the micelle was simpler, so I'm a little surprised that like the vesicle didn't fuse with the micelle and the micelle.
I think the bilayer is actually more stable.
Yeah, you're right, I mean, that's what it looks like.
Okay. Okay. So here, see if you understand. Which catalyst is it better for a vesicle to have? Is it better to have SH or is it better to have SHG? In this one, they put SH and SHG in the tubes, mixed them together. So what do you think? Point up if you think SH is better to have, point down if you think SHG is better to have. Okay, so everybody's got it, right? It's better to have SH. SH outcompetes SHG. In fact, if you put them together, SH grows and SHG shrinks. In a sense, SH is eating SHG. Predation. Okay? Now, if you think about it, in the primordial soup, there probably were lots and lots and lots of different vesicles with lots of different things inside them. And so you had a really complex, diverse environment. So they wanted to see, okay, what happens if you put all three of these things together? One with one catalyst in it, one with another catalyst in it, and one without. And you can see clearly SH gets a gold medal, silver medal, and in evolution then you get a bronze medal. Okay, now another cool thing about vesicles, typically they're spherical. That's like the simplest way they can be. When they consume micelles, they grow faster in surface area than they do in volume, and that makes them become filamentous. If you gently shake them, like maybe would happen in a hydrothermal vent as the current goes around, they fragment into smaller vesicles. So this is akin to cell division, right? Okay, now, another cool thing, when they grow, they actually generate a pH gradient across the membrane. The chemistry of that, you read the paper if you want to know, it's a little bit complex, but they'll generate a pH gradient across the membrane. And where do we find pH gradients across membranes? Mitochondria, chloroplasts. Right? So this is a fundamental basis of a lot of our energy processes in cells. And so in these protocells, you can get gradients, useful energy gradients. So sum it all up. What do we got? We got a barrier that we can create under primitive conditions. We got, we don't have an information molecule. That's the topic of another paper. We don't have time to really talk about that. But we got something that can grow, right? It eats other micelles, it eats other vesicles. It can reproduce. You shake it, it fragments into little pieces. It can metabolize. It can do catalysis, which is part of metabolism. It can create that pH gradient. And it can also compete, which is necessary for evolution. So in this tiny little thing, yeah, it's not alive because we don't have RNA to be the hereditary molecule. But where do we go from here? The next thing to do is add a self-replicating RNA, which actually we have in another group was doing this. They got a self-replicating RNA. Get that RNA ribozyme to give the thing competitive advantage and somewhere down the line create life. Any questions? Please, questions.
So, the previous slide, all those characteristics, the division,
Everything in red.
the ability to eat basically other vesicles, that was all because of the SH tag.
So because it had the catalyst SH, it was able to compete more than others, better than others. I mean, vesicles alone will uptake micelles.
Okay, I believe that.
Okay? And maybe that's kind of a primitive eating, but you wouldn't call a vesicle with nothing in it alive. I mean, it can't replicate itself. It can fragment, but there's no metabolism. So,
Is it the SH itself, or could it have been another catalyzing enzyme?
so SH worked. SH, what was the other one? SHG. So yeah, both of those were ones that actually gave it, a competitive advantage. So it could be, it could have been an RNA catalyst doing the same thing.
Why when both vesicles had SH, there wasn't growth?
What's that?
Why when both vesicles had SH, there wasn't growth?
Okay, so think about this. If you are cloned and you're competing with yourself, do either of you have a competitive advantage?
Got it. Yeah.
Right.
What is the SH actually doing that allows the vesicle to eat other cells?
So what the SH actually does is it takes in a food source, and because this localizes to the membrane, it actually makes this membrane better able to uptake fatty acids. So that's the energetically what's happening. SHG is a slower catalyst for the same process. It would have been nicer if peptide, a tripeptide is better than a dipeptide, because then you're moving toward more complex protein. I mean, it's not really a protein with two amino acids. But. Anything else?
So you're considering organisms that are basically sacs with no information in them or intelligence not to be alive. What are you getting?
Well,
Wow, dude.
you should feel insulted, I think.
Yep.
I'm just
Just
clarifying
saying,
so people, objects with no, that just sit.
Well,
I really like
it can still be alive even though it has no intelligence. Right? It just has to be able to grow, reproduce, and eat.
Very bad joke.
All right, thanks a lot for coming.